Good afternoon, everyone. Thank you very, very much for coming to this keynote speaker that's part of the HEMONC Ground Rounds seminar series. It's also a continuation of our Talbot family pancreatic cancer awareness reception that we had last night in honor of the Talbot family. Jane Talbot is in fact here again today. It's great to see you, Jane. Just a little extra plug for Jane. She's no longer leading officially the South Carolina affiliate of PanCan. She's taken on more of a voluntary role. So she decided to take a step up, I guess we can say. And now she's officially part of our PRC committee at Hollings, which reviews our IRB protocols as is a patient advocate for that. So let's give her an applause. Constantly learning, constantly helping. Thank you, Jane. So it is indeed a great pleasure to introduce Dr. David Tuveson as our keynote speaker, who is the Cancer Center Director at Cold Spring Harbor Labs. I'm not going to take too much of David's time, but I do want to give you a little bit of background. First is his academic record. So he got his Bachelor of Science from MIT, even though he's originally, he told me, from the Midwest. Most of his career has really been on the East Coast. And then did an MD PhD at Johns Hopkins. During his PhD portion in Doug Fearon's lab, he only published first author in Science and a JX Med paper. So I think that was pretty successful for a PhD scientist. And then after completing his MD, did internal medicine residency not at Dana-Farber, but at Brigham's, and then did a fellowship in medical oncology at Dana-Farber, and then pursued that with a postdoc back at MIT. So he went back to MIT for doing that, and then started his own academic career as an assistant professor at University of Pennsylvania. After finishing that assistant professor, he got an opportunity to go overseas and was essentially promoted, if you will, to associate professor at Cambridge. So, and I think led a pancreatic cancer group there. And then came back to the States, full professor. He took over as the deputy director at Cold Spring Harbor Lab in their cancer center. And then 2016 was promoted as their cancer center director. So it's going to be 10 years next year. Quite an accomplishment. I can tell you that besides renewing their cancer center core grant, he just recently, this last year, got a new R01, so very active in research still. And I'll only say that there are many, many, many pivotal papers that have been published. Those of you in the pancreatic cancer field, of course, know about the models that he's produced, like the KPC-GEM model, and of course, all the cell lines that we've all utilized for orthotopic models, but also elegantly the organoids that he's also made for these orthotopic models to really recapitulate the microenvironment and also the progression of pancreatic cancer, which is really what really is the most important phase, we think, of this cancer. In addition to that, learning about NRF2 and how it buffers the reactive oxygen species, and also important for translation, and also the CA19-9 as a marker, as we know, in pancreatic cancer, and then appreciating in a mouse model that really drives this disease. All these papers published in Science, Cell, Nature, and Cancer Cell. And I'll end by saying that two things. One is that with all these accomplishments that I just listed, still I think we know you best, David, as the work that you've introduced to us with the cancer-associated fibroblasts, and being able to appreciate their subtypes, the heterogeneity, their different functions, and whether they control fibrosis, whether they control inflammation, or even antigen processing. It's just been marvelous to be able to follow what you do and how that impacts our knowledge of pancreatic cancer and hopefully our future treatment. And also just to recognize that all of us who run labs and who train trainees, the fact that you've trained over 40 postdocs and 20 graduate students is really a credit to what you've meant in the field as a mentor as well. So thank you very much for coming. I know this is a repeat visit to MUSC. I think you said you'd been here about 10 or 12 years ago before. It's really a delight to have you here as our keynote speaker for our Talbot reception. So please help me welcome David Tuveson for his keynote talk.
All right, thanks very much, Dennis, for your generous introduction. And welcome again to Mrs. Talbot and their family, and thanks to them for sponsoring the Talbot Lecture. So in the next 50 minutes or so, I'll try to talk about a couple of unpublished stories from our laboratory. Perhaps get you to think differently about pancreas cancer, and maybe for those of you who don't work on pancreas cancer, you'll feel jealous and you'll join the groups that work on pancreas cancer. And that last picture is going to be informative today. This is my disclosure slide, which is highly relevant for today. So the field of pancreas cancer research is thriving these days. There's lots of people with ideas. There are a lot of trainees excited about it. Companies are now working on pancreas cancer, and they used to not want to work on pancreas cancer because all our randomized phase 3 trials would fail. And so now their companies are alert and they're back. Pancreas cancer is no longer a rare disease. We now have more than 60,000 patients diagnosed in our country per year with it, and we lose most of them in that year. Nonetheless, the survival rates have increased incrementally over the last 2 decades. Part of that is because patients are getting treated. So if you went back 20 to 30 years, about half the patients got no medicine. Now that's uncommon. I would say 80% get some medicine. When I say some medicine, I mean some medicine to fight the cancer, not just medicine to help with their suffering. So we've made some progress, but not our big splash, not yet. Over the last couple decades, we've spent time trying to figure out what is pancreas cancer. And we've characterized the tumors from our patients. We've developed model systems to help us dig deep. And we've learned a lot from that. So we know a fair bit about the genes that are mutated or misregulated. And because of that, we know about the biochemistry, the pathways, et cetera. We've given names to the precancers and the invasive cancers. And we've started to learn that link between a diseased pancreas and a neoplastic pancreas. And so your pancreas, again, is this organ located behind your stomach. It's in a human, kind of the shape of a banana. In a mouse, it's not quite as anatomically pleasing, has many more lobes. But you really don't think about your pancreas. You can't feel it. You only think about it if there's a problem. And there's a problem if it hurts, or the problem if your blood sugar is not balanced. And so because of that, it's been this dark organ. It's a vital organ. You can't live without it. But we've learned a lot about the pancreas over the last couple of decades. And those of you who study the cancer are actually helping the folks who study the health of the organ. And last night, when there was a meeting in this room, which was more about advocacy for pancreas cancer research, it was led by the family, Ms. Talbot's family. And I talked a little bit about those topics I wish people would work on, and I'll mention some of them at the end. But the last line is one thing that you all are actually world leading on, and the systemic illnesses of pancreas cancer patients. People are sick when they have pancreas cancer. They hurt, and they lose weight, and they become unfit very rapidly. So they don't actually qualify for clinical trials oftentimes. And actually understanding the systemic illnesses of pancreas cancer, be it the wasting syndrome, the cachexia, the clotting syndrome, the Trousseau syndrome, et cetera, are important. And fortunately, we know more about both of those illnesses, and we have ways to actually start to interrogate them, thanks to the basic science, some of it coming from actually Dennis's lab and his colleagues. This is a cartoon of pancreas cancer. Every cancer has a cartoon. This is ours. You have a normal duct on the left, and even a normal acinar cell. These are 2 epithelial cell types in the pancreas. And on the very right, you have cancer cells that spread through the body. And you have the various mutations that we think occur in some stepwise fashion. Every cartoon is simple and partially true. It turns out that most of us, some of you are hopefully too young to have this, but most of us who are, let's say, over the age of 40 will have mutations of RAS in our pancreas. And most of them will do nothing. They'll just sit there. Sometimes the cell will continue to acquire mutations, and those cells can go on and invade. These days, the math is around 1 out of 50 people get pancreas cancer. So it's not super rare, and that's too often. And so trying to understand why only 1 out of 50 get cancer, but 50 out of 50 have RAS mutations in their pancreas cancer is just a fundamental question that could be asked. Should you need a thesis topic? What we have learned from modeling is that this KRAS mutation I mentioned is pretty strong. And so if you put in a KRAS mutation into a cell or put it into a mouse and express it in different tissues, you actually oftentimes can make a hyperplasia in the lung, in the intestine, in the pancreas. And you can start a process that would lead to not just a hyperplasia, but a dysplasia, to the point where a pathologist would say, that looks like a dysplasia we have in humans, which we call pancreatic intraepithelial neoplasia. And so RAS is sufficient. And we made a mouse that has a spartate mutation at codon 12, and we've now made one that has a valine mutation, and they both, can do this. And if you wait long enough, they actually will develop all by themselves an invasive cancer. And so, although that's true, that it's sufficient to develop the disease, it is a very inefficient process. And what I mean by that will become graphically relevant when I show you pictures of thick sliced sections of the pancreas where we're counting the number of precancers in a mouse pancreas. So it's wildly inefficient, the process, which is good. So cancer, again, is a pathetic disease. The only reason we can study it is because it is so profoundly pathetic. Think about that. People talk about cancer is tough, rough, strong. BS. Cancer is like the most deranged and deficient disease process I'm aware of. So we have a shot to beat it. It's like a wounded enemy. And actually, a lot of our drugs just like expose the wound, okay, because the system is poorly designed. And so since it's sufficient to initiate, but inefficient to progress just by this RAS mutation, you could ask why. And one is these things have to happen around the cancer. Inflammation, for example, which is a topic we're quite fond of. And it can also be there's a rare cell that stochastically acquires the right assortment of molecular events to become a cancer cell. And that's possible. Could have progenitor cell activities. And there were some really interesting papers from Scott Lowe and Tuomas Tomela about that. Well, what about the possibility that it's not just the neoplastic cell, but it's those cells around it that really push the process? So we have this overarching hypothesis that all by itself, the epithelial cell is not going to do anything. It needs the surrounding stroma elements really to make that decision, to overcome a threshold of events. And the specific hypothesis is the fibroblasts, which are really the peacemaker cell of every tissue of your body, becomes the dark peacemaker, becomes the cell that actually puts into motion a cascade of events, which leads to this productive output of a bad disease. So that's our hypothesis. I always tell my trainees, I'm like, tell me what problem you're excited about. And they say this and that. That's okay. What's your hypothesis? And they say, well, what is your? I'm like, I got a lot of hypotheses. You got to tell me your hypothesis. I don't want to infect my hypothesis on you. So own a hypothesis, but be equally happy discarding your hypothesis, because your job as a scientist is to disprove your hypothesis, not to prove it. Don't ever act like that. Try to kill your hypothesis, because then you will set up the most rigorous experiments, because you'll have the best controls. And along the way, a lot of times your control ends up being way more interesting than your hypothesis. So in any case, the specific hypothesis I'm going to try to disprove to you is that the fibroblasts are playing a role in this. So how we got into studying the fibroblasts is kind of a long story, and I'll just give you a snapshot of it. We were trying to study it in the animal models and in the patient specimens. Really hard. it's hard to get the cells out and to ask them questions. They get tired fast. And we could never figure out the conditions to make them happy enough to survive in culture. But when we got into the organoid technology space, due to a friendship I developed when I lived in Europe with Hans Clevers, we developed the organoids in the mice and in the human samples. It turned out if you mix precursor fibroblasts from the pancreas, which are buoyant cells that have all the properties of so-called stellate cell of the pancreas, immortalize them, still act like stellate cells. If you mix them with the epithelial organoids in a three-dimensional matrix, we used a commercial product called Matrigel, but you can use lots of different ones. The fibroblasts get excited finally, and they get excited, they make matrix. And so here's a picture. Oh, yeah, I got this pointer thing. Okay. All right. So this is actually made by the fibroblasts. So these are H&E stains of Matrigel drops that either just have the organoids, which are epithelial cells, or they have the organoids with the stellate cells and see all this pink stuff by H&E. And the same, section below, you can stain it with Mason's trichrome, which obviously pick up extracellular matrix proteins, mostly collagen. And so we were making desmoplasia in a drop, which was, cool, because we had never figured out a way to do that before. And then we studied the cells that we started with, and it turns out the cells we started with became two different types of cells. Ones which had myofibroblastic properties, could make matrix, activation of the TGF-beta pathway, all those, types of functions. Another population, though, were not like them. They didn't seem very interested in making matrix and did not have a TGF-beta pathway gene signature. Opposite, they acted like immune cells. They secreted IL-6 and all these other cytokines and chemokines and interleukins. And we thought, well, one cell can become two. And it turned out if you took these cells with this property and plated it differently in a high tension setting, they became myofibroblasts, and you can kind of toggle them back and forth. And so that led to a series of observations using tissue and other methods that suggested that there were these two populations of cells. And it turned out there was another population by using the early versions of single-cell transcriptomics, which had fibroblast gene expression, but didn't have those two properties I just talked about, but rather expressed class 2, the molecule that antigen-presenting cells use, and had CD74 in class 2, lacked co-receptors, so probably couldn't be a dendritic cell. But these were cells that we could see in the pancreas, we could find it in the human, find it in the mouse, and suggested maybe they had some immune regulatory function. And so we started out by describing these three types of cells, and here's some pictures from those papers. Since that time, so our last paper was like a long time ago, I don't know, 6 years ago, seems like 16, on this. And since then, it's been great because lots of really smart people have jumped in and done their piece on trying to study these fibroblasts, and they've given them alternative names or additional names. And every time I go somewhere, people say, well, how do you reconcile your findings with the field as it's emerging? I said, well, we will keep working. And you'll hear about some of that work today. And so the matrix, though, that's deposited, you see, and this is a mouse tumor, and these are all mice tumors, I guess, does a lot of really interesting things. It's that packaging material. And so you would say, well, maybe it does some physical things to the tumor. That packaging material actually serves as a nutrition source for the cancer cells. The cancer cells eat the matrix. That's how they derive glutamine, for example. But it's not a packaging material that you would want if you receive something from Amazon, because it's more like cement. It compresses the vasculature to the point where the vasculature is poorly functional. And so these tumors are oftentimes hypoxia. We think in part because the matrix is compressing the vasculature. Now, if that's the case, that would mean a couple of things for the cancer. One is they probably don't get enough food, sugar, amino acids, lipids, et cetera. They don't get enough oxygen. So these cancer cells are not going to be happy because they're going to have all these gene expression programs to survive in the setting of that type of environment. And if you're a therapist, you're trying to get your drugs into something like this, you're going to have a hard time. And so these are just, some fundamental, points. And the last one is the immune system. We're all excited to get the immune system to clear out the cancer. Well, the immune system needs a lot of TLC for it to work. Has to have a lot of oxygen, has to have enough sugar, has to have enough glutamine, or else they're just not going to work. And you can call them whatever form of exhausted cell you want. It's kind of irrelevant if they don't have lunch and breakfast and dinner. And so these tumors in the pancreas they're a pretty hostile place for really anything that you would like to do as a therapist. And so again, we studied this process very carefully. We started with wild type healthy pancreas, which you see on the left. We compared it to inflamed pancreas, which is in the next box here, and early cancers and advanced cancers. And we did the single cell approach. We had to first learn a way to digest the normal pancreas in such a fashion that nothing died. And for those of you who work on the pancreas, you have appreciated that when you try to manipulate the pancreas, it can get kind of messy fast, because the acinar cells in the pancreas, boy, they're really good at digesting things like their neighbors. They make all the enzymes to digest RNA, protein, DNA, fats, carbohydrates. They are the best source of enzymes in your body. So if you're trying to get viable acinar cell cultures, a lot of people say, well, what other projects do you have in my lab, sir? It's hard. So we have this terrific postdoc who's now gotten promoted twice in 5 years. And he's now called assistant professor in the medical system that we collaborate with, Sandeep Nadella. And he figured out a way to digest the mouse pancreas on ice. And so we use enzymes from bugs that grow on ice. And it actually works, not really well, but works well enough to the cells that you get out of it are all viable. And so you get viable acinar cells, viable vascular cells, viable fibroblasts, viable, this and that. And because of that, we were able to go deep on what are the resting cells from the pancreas. And that was very important for the two stories that I'm going to talk about today. I'm actually not going to give you a whole Sandeep's lecture because it's not quite finished, but it'll be cool when it's done. And that'll be before 6 years from now. And so we get this data from single cell, and the data looks like the stuff on the left. It's very sparse. So single cell is great, but it's very superficial. It's kind of like proteomics. You only learn so much. So you have to have very focused questions so that you get much more robust data. And that's what's shown on the right. So if you take the same data on the left and you apply a computational method to the data where you're looking for pathway activation as opposed to presence or absence of some gene, you actually learn more about the cells that you're trying to describe. And if you look at the shape of those boxes on the left versus the right, it's not exactly the same. So on the left, you have like fat box, a skinny box, a fatter box. Look at the brown and look on the right. You're looking at those three red boxes. They kind of look similar in shape. That means you reclassified cells just by using the open UMAP kind of approach. So we've been working with Andrea Califano, who's a really great computational scientist at Columbia for about 7 years now. And this is the approach that we used in Ella's paper 6 years ago. And we've applied that to that last slide where we had the 4 states of health, inflammation, early cancer, advanced cancer. And taking all the data from all the tens of thousands of cells, this is how you can summarize it all. And so we have clouds of pathway activity. And looking again at the fibroblasts. And so on the left, you see the normal pancreas. Those fibroblasts have a sort of West Coast activity, 9 o'clock. And if you look at the pancreatitis mice, they overlap that, but then they have more of a 12 o'clock button too. And if you look at early cancers, it's overlap with chronic pancreatitis with trending clockwise to 1 o'clock. But look at the tumor fibroblasts. They are a different beast altogether, very starkly different. And the 3 types of fibroblasts I talked about before, you can actually look at all of them in this kind of genre. And really, where you look for the cancer fibroblasts, that's where you see their big bunch up of the myofibroblasts, the MyCAFs. And I'm going to talk now in 2 vignettes about that. So of the 4 types of cells, you can mathematically separate them into really 5 cell states, 1, 2, 3, 4, 5. And they're shown here, mathematically as probability statistics where you see cell state 1 and 2, and here's 3, 4, and 5. And the normal fibroblasts really fall into the cell state 1 with some in cell state 2. Pancreatitis, which is CP, you start to see that, which is a green curve, having both cell state 1, 2, but also start of cell state 3. And then cell state 4 and 5 really are occupied by precancer, which is KC, and the advanced cancer, which is KPC. And that was kind of interesting because the precancer, which is KC, is the one that has the most interesting mathematical probability curve. So you have cells that do overlap with advanced cancer and cells that overlap with inflammation. And so if you look at the pathways, though, as biologists, it's easier to not think in the abstract, but in the, the pathways that you know about, that you read about, that you care about. Look at, so here's a rendition of some 20 to 30 of these pathways. And you look at the cell states on top here. Not really sure how this thing works. But if you look at the cell states on the top, you can separate, like put the Mississippi River down the middle. On the left, you have inflammation and normal, and on the right, you have cancer. And so you can just ask, like, what's happening in this space in the inflammation area versus the cancer area? And so on the very top, the first arrow is at a gene cluster, which is talking about the nerve growth factor receptor pathway in fibroblasts. And some of the genes are listed there. And below it is TGF-beta, the other arrow. And again, I don't know if you can appreciate this so much because you have this fancy light thing. But you're supposed to appreciate these little red dots, which are basically just looking for the TGF-beta transcript that you see in inflammation and in the various cancer types, but not in the normal. So let's just take that as a, quote, positive control. But thinking about the nerve approach is going to be the first vignette. Here's a cartoon of how people study nerves in pancreas cancer if they're an experimentalist. So this is Tim Wong and Alec Kimmelman. Tim had a pair of papers talking about how sympathetic nerves promote pancreas cancer. If you use poisons, he could prevent it. If you use scissors, he could stop it. And the parasympathetic on the left-hand side, kind of opposite. He thought that the parasympathetic nerves through the muscarinic acetylcholine receptor pathway would prevent pancreas cancer by secreting acetylcholine. And on the bottom, Alec Kimmelman said the nerves feed the cancer, feed them serine. He had a very provocative paper about that. So again, this is in mice. In the humans, what do we think about the nerves? Well, we think the nerves are why we have pain. Patients present, if they're lucky, with painless jaundice. Eyes are yellow. They feel fine. Their family says, you don't look good. You go to see the doctor. And if they're lucky, they see Dr. Hawkins. Dr. Hawkins says, you have a tiny tumor. I can schedule for surgery next week. Now, that's a tiny fraction of the patients that present like that. Most people, they don't feel right. They're losing weight. They have pain. What is the pain? Pain means there's a nerve that feels something that's not supposed to be there. You know what pain is? It's like when you burn your hand or you smash, stub your toe, or you eat some hot stuff that you don't like. That's pain. Well, how do those kind of nerves work inside your pancreas? So again, sensory nerves sense pain. Those nerves are not sensory nerves you're looking at from Tim. Alec's nerve is a sensory nerve. That's a sensory nerve. It's feeding the mouse cells, cancer cells, serine. We looked at all of this stuff in the models that I just talked about. So on the upper right is a little cartoon of a model. You can take these stellate cells from the pancreas, stick them in Matrigel. They're pretty quiescent. They're not activated. They'll just sit there. You just have to feed them. They won't go anywhere. They're not going to proliferate. But if you give them TGF-beta, you can kind of promote them into a process where they think they're a myofibroblast. If you give them interleukins, particularly interleukin-1, you can promote an inflammatory behavior. You can also use conditioned media from organoids and get that to happen. So you can compare these two states kind of trivially by transcriptomics. And when you do so, the top process you get in these myofibroblasts is actually axon regeneration. You would have thought we would have figured that out 8 or 9 years ago because we did the first work on this, but somehow we just missed it. So it turned out that is like the top hit pathway that when you do a comparative omic process, you would see. And if you're happier by Western plots, I find them much more pleasing than to lists of stuff myself. If you give TGF-beta to these stellate cells, they make nerve growth factor. And some of you would have remembered NGF because it was like the classic axon guidance gene of central and peripheral nervous system that you learned about when you were young. And a lot of these other proteins will show up too, like fibronectin and semaphorin, various types. And it turns out having that receptor to the TGF-beta receptor is necessary for that biology, because you can knock it out in organoids and you won't see any of that. So again, these are models from cells in a dish. And if you do a similar type of experiment where you take the celiac ganglia from newborn mice, stick it in a dish, and then take the media from the cells that you either have given TGF-beta, that you give TGF-beta to, but the stellate cells either can sense or can't sense it, because you could knock out the receptor. So you add all that cocktail stuff to the cell, so it still includes TGF-beta. I don't know if you can appreciate this, but there's a lot of hair here. These are axon spikes that are coming out of this culture of celiac ganglion, and it's a lot less here, even though you see these big cells. These aren't actually the axons sticking up. These are the cells migrating away. And so you can promote axon outgrowth by using this conditioned media, and the conditioned media that has the activity has the NGF and other factors as shown here. So again, this is in vitro stuff. So I promised you before that I was going to show you a graphic picture of how RAS is sufficient but inefficient. So these are thick slices that neuroscientists really taught us how to do in the brain, where they could get pretty structures of how nerves were connected and how the cells around the nerves played a role in the spatial architecture of the brain. You can apply that to other tissues in the body. And so Jeremy Nigri, the really terrific postdoc who's been driving this project, did so. And the normal is on the top and the early cancers on the bottom. And the red here is the stain for cytokeratin 19, which is, as you know, a protein that is expressed in ductal cells of the pancreas. Tubulin 3 is a pan-neuronal protein. So you're looking at nerve tracks here. Protoplanin is a marker of activated fibroblasts. And so you have a little patch of that. It turns out this is the outside of the normal pancreas. You can add them all together. What if you look in a mouse that has these early cancers? Now, mind you, this is a space that's, you know, like millimeter by millimeter. Look how many different CK19 positive little bumps you're looking at. These are all the precancers in the mouse. The mouse has millions of precancers, millions. And where you see the bumps, the nerves have gone crazy. And where you see the nerves, you see the fibroblasts have gone crazy. So it's like saran wrap around cauliflower, and the cauliflower has some wires around it, if you like metaphors. What kind of nerves are we talking about? Well, this is where, you know, it's not hard to actually ask that question, because they're very good protein markers of the various nerve classes. The one that really matters is actually tyrosine hydroxylase, which is a sympathetic nerve marker. And so where you see red, you mostly see green, and you overlay it, you see mostly yellow. You have a few red, and these are the sensory nerves, but they're way outnumbered by the sympathetic nerves. It's not even close. Can you do any of this in the human? Well, some of it. And so you can make primary cultures of stellate cells. You can immortalize them from organ donors, which is, you know, really how you would get them. These are people who usually are in motorcycle accidents. And there's two examples shown here, and if you treat them with TGF-beta using basically the same methods I talked about before, they also turn on production of these various proteins, including pro-NGF, which is the precursor to NGF. And if you look at the secretome, meaning the fluid outside the cells, you then see NGF there as, you know, up in both. So it looks like in the human you can see some of these things in vitro. And how do you do the celiac ganglia experiment in the human is not, you know, non-trivial. And so what we use is a model system, which is the rat PC12 cell, which is a historic cell line for those of you who are, you know, cancer research historians. This is the cell line where we studied what does acute versus chronic stimulation of the MAP kinase cascade do. And chronic usually leads to neurite outgrowth. And it turned out the media from cell lines, which were treated with TGF-beta, the human cell lines, actually also promoted axon outgrowth of this cell line. Again, suggestive that the processes may be similar. If you look at human tissue, we haven't been able to do the thick slices of the human tissue. For that, we have to get a, you know, essentially a patient that was in the OR that has tissue that would come to us and not to pathology. And even though the pathologists are very friendly with us, occasionally the health systems are less so. And so we had to use paraffin sections to actually ask that question. And here we used a different protein marker for nerves, which was peripherin. And you see the peripherin here or near the cancer cells, which are red. And here's another example where you see the peripherin stain, which is in the green dots. And again, here's your cancer cells. It turns out the green dots are in the white, and the white is where alpha SMA or the myofibroblasts are sitting. Again, correlation, but suggestion. What about experiments? And so I said before, Tim actually already shown that if you cut the nerve or poison the nerve with sympathetic toxins, you can actually change the biology. And here's our foray into that. We took tumors in mice, we transplanted some cells and organoids into mice, made some tumors, and used a poison for sympathetic nerves, which is 6-hydroxydopamine, and asked what would happen. Well, if you use the poison, it's less blue when you stain for Mason strichrome. You're making less matrix. And it turns out the tumor is smaller too. So it seems to have an anti-cancer effect at first approximation, essentially confirmatory. If the hypothesis is correct, then that means the fibroblast is, and it makes NGF, well, then the fibroblast has that possibility, if it's the beacon for the nerves, of responding to the nerve in the first place, right? So what do sympathetic nerves do? They make catecholamines. The chief one they make when they're sympathetic nerves and innervating is norepinephrine. And well, how would a fibroblast respond to norepinephrine? Well, through the adrenergic receptors. There are several of them. There's the beta receptor, which is very well studied, and then there are alpha receptors. And there's different betas and different alphas. Well, one of the ways that cells can respond to neurotransmitters is they can flux calcium. And we asked, is that the case? So Jeremy put a reporter for calcium in the fibroblasts and applied neurotransmitters, including norepinephrine. And this is a still shot of an actual live movie thing. And if the vehicle looks like this, if you apply norepinephrine, you flash calcium. And if you use a blocker to one of the adrenergic receptors, you essentially blunt all of that biology. And that blocker is a blocker of alpha, specifically of alpha 1. And that stuff is, kind of summarized in the bottom, and there's your various tracings here. He looked into asking, could the betas do it? So there's a lot of ways to do it. You could use beta blockers. You can use specific beta agonists. The answer is no. Beta did nothing, in this space. And then he asked, well, if this is the case, then they have to have the alpha receptor in the first place. And this comes back to the point I was trying to mention to you before. We basically don't see the alpha receptor by single cell transcriptomics. It's expressed too low, so you don't see it. Well, it doesn't mean it's not there. So again, don't draw too many conclusions from any of the single cell kind of experiments you're doing until you have orthogonal methods. So again, using alpha as the protein, you can see alpha on the outside of the normal pancreas, but not on the inside. But when you have inflammation of different kinds or cancer, you start to see alpha, which in this case is colored red, I guess. So, and it's in the cells which are green, which are protoplanin sensitive. And so, just imagine this is a red and green cell because I have the white arrowhead pointing to it. And you only see it in the setting of the cancer and the bad inflammation. If you look in the human, it's pretty similar. It's around the outside of the normal pancreas, but on the inside, it's basically in these cells that are SMA positive, not really in the cancer compartment. And so again, consistent with it being present so that it could mediate the biology we're talking about. Downstream of alpha, there's biochemistry of different kinds. There's activation of the MAP kinase cascade, but most importantly, phosphorylation of a protein transcription factor called CREB. And you can see evidence of CREB phosphorylation after 30 minutes of phenylephrine, which is a synthetic analog of norepinephrine, somewhere here. So here compared to there, and in the various Cath lines, you see the same thing. And you will also see variable activation of the MAP kinase cascade. So again, consistent with the biology that we think of from that receptor. And if you look at the three big receptor classes, alpha 1, alpha 2, beta, beta 1, 2, these are the pathways they usually activate or inactivate. So alpha activates phospholipase C, which cuts PIP2 into IP3. and diacylglycerol, IP3, as you know, due to the work of Solomon Sider, binds to the IP3 receptor on an internal calcium store. It's sort of it's a membrane bound organelle where calcium is stored inside cells and then is released. Alpha-2 does something else, actually inhibits calcium fluxes, and beta doesn't do calcium at all. It's a cyclic AMP activator by activating adenylate cyclase. But as you've seen, the sort of theme that I'm trying to introduce here, the neuroscientists were way ahead of us in studying these receptors because they could generate recombinant versions of these systems where they could shine light or put in synthetic drugs to activate or inactivate these various systems. And so we use the latter, which is a chemogenetic system where you took the alpha-1 gut and stuck a different thing on its amino terminus to bind to a synthetic ligand. And this is the DREAD-based system. And so if you add DKZ to it, you could activate the system synthetically in a cell. And again, the experiment that we were hoping to do was to put this into fibroblasts and see if that would do anything. And the way we did it was to actually use a system where we could express it as a recombinant knock-in. And so the DREAD system is the purple guy right here, and it would not get expressed until the lock-stop-lox cassette is removed by adding tamoxifen. And we used the PDGF receptor alpha as the common fibroblast-like gene that we have found through our single-cell work to be present in all the various fibroblast types that we ever found in the normal pancreas of the mouse and the human. And so the experiment was to activate expression, test it in the setting of inflammation and in cancer to see if by hyperactivating this system, by giving this synthetic ligand, would that have any impact on the biology we're seeing? And so the first one was just inflammation. If we give the DKZ in a system that's competent to receive it, in the setting of cerulean, the mice lost a lot of weight to the point where the vet was calling us up and saying, hey, you got to stop this. It's off the IACOC protocol. I'm like, okay. So we did a week. They lost a lot of weight. And we got the tissue. And here's what the tissue looks like. And so again, this is pancreatitis for a week or pancreatitis plus DKZ, and you can't really appreciate anything because I can't. But it's a lot of dots here compared to a few here. Let's go over here. Essentially, you have a lot of Mason's trichrome compared to here, and a lot more podoplanin here to here. You have a lot more TNC, which are, again, the activated fibroblasts that are SMA positive, TGF-beta responding. Tons more fibroblasts, macrophages, and the macrophages are activated. They're angry. They have arginase 1 expression and other things. So it seems like we got a lot more inflammation. Mice lost a lot more weight. What about in the setting of RAS mutation? So it's a similar question, but for this, we collaborated with Mike Ostrowski, who was generous to provide his system, which was a PDX FLIPO insert knock-in, where the flip, again, is different from Crelox, sees a 48 base pair sequence, and it could loop out, in this case, a stop cassette upstream of RAS, similar RAS mutation of G12D. And so the mouse would have this, and then when we added tamoxifen, we could turn on that other system of DKZ, of chemogenetic activation of alpha-1. And so it was kind of a similar experiment to before, other than it's in the setting of the RAS mutation. And in this case, we carried it out almost for 3 weeks. And every time we gave a shot of the DKZ, the mice would lose weight, get it back, give another shot, lose weight, get it back, lose weight, So they were responding to the DKZ like they did in inflammation, but they could recover, from it each time. So in the end, it's the same kind of deal. We got the tissue back. What did the tissue look like? Again, harder to appreciate given my lack of high fidelity pictures for you all, but this is the control, and this is in the setting where we have Mike's dual recombinase system. You have a lot more blue. The most relevant thing is we have a lot more high-grade PANETs. So it promoted progression of the pre-invasive cancer. We're still trying to get it on the setting of the RAS mutation and the P53 mutation, so I don't totally have that data. We had to do a kind of end run to do that, where we had a transplantation model in the setting of the DKZ, but when we do the transplantation model, you get more blue and the tumors are bigger and the mice are sicker. So it seems that the system, actually promotes the disease. And so again, I said before, the nerve shows up, we think, because the fibroblast sends out that signal. When it shows up, the nerve has the possibility of releasing its payload, which in this case would be norepinephrine. We think what's happening is that the initial insult is that the cancer cells are releasing TGF-beta as the latent form. It gets cut in the, to be active. When it becomes activated, you can make a myofibroblast. Myofibroblast, as part of its first thing, is to release axon guidance cues. Sympathetic nerves come in. This is in the pre-cancer, pre-invasive, PANIC. When they show up, if they release norepinephrine, they're going to hyperactivate those same cells. And those same cells, when hyperactivated, mean the myofibroblasts, now will really promote the rest of their activity, which is to get the monocytes to come in and to promote a bidirectional crosstalk with the cancer cells. And so, you all possibly have heard of perineural invasion, PNI. This is something that on the pathology report of a patient, you get that information. That's related to pain. This is different. This is invasion by the peripheral nerves. This is IPN, okay? But it's not the sensory nerves. It's the sympathetic nerves. So we think this is probably the first wave, and then, that's our hypothesis that we have to now disprove. So that's the first story. The other side of the Mississippi is the second story. That's the cancer space. When you look in the cancer space, there's a lot going on, but it turns out there's one pathway that pops up twice, which is the Wnt pathway, which is not a new pathway. It's required for biology and development between endoderm, mesoderm. People who study the intestine think about Wnt. Pancreas, less. It's not mutated. Of course, it's relevant for all the organoid culture work. But we saw Wnt. Didn't think much of it, but then we started to look into it much more critically. And it turned out, when we did so, if you compare cell state 4 and 5, again, this would be like the early state in invasive cancer, the advanced state. Cell state 4, you're seeing canonical Wnt signaling massively. You see negative regulators of Wnt like NKD2, which is a marker you'll see many times in the next 10 minutes. Cell state 5, you lose that. And actually, you're starting to look at pathways involved in tension, row, rock, lots of things in contraction. And so we looked for these mediators in this tissue using in situ hybridization, which is a very beautiful and tedious and expensive way to characterize biology. But if you have trainees who are patient enough, they can give you, well, pretty images. And so baby blue is NKD2. This is a single gene marker of activation of the Wnt signaling cascade. You see baby blue cells outside of the epithelial cells, sometimes when you have an early cancer, all the time when you have an advanced cancer. And the other arrow are for a TNC positive, which would be the cell state 5 cells. In the human, we basically see the same thing. We're using different marker genes for the ISH, but by single cell, et cetera, we see the same markers. This is an advanced human cancer. So with this knowledge, we then could go back and revisit how do you make desmoplasia if this was involved. Because again, cell state 4, we see Wnt activation, and I didn't say it, but we see massive production of matrix factors. Matrix genes are highly, highly upregulated at that stage. And so we asked, if you took a model system where you just had those stellate cells either in matrigel or in some type of gelatin, bed, and you asked, what are the factors you need to add together to get desmoplasia in a dish without having epithelial cells present? So cocktails of Wnt and TGF-beta, either by themselves or combination. If you look on the very right, you see a lot of red because you have a lot of gene expression that myofibroblasts have when you add these cocktails together, that you don't have one or the other. And so with this result, we then could really take a page out of Eddie Kuckerman's book. And so we learned from her how to study fiber formation in vitro, which she had done some really beautiful work learning how to measure anisotropy of fiber formation in vitro. And it turned out, again, when we used the two together, we got lots of parallel fibers that by her level of analysis is, kind of the most robust fiber formation that has ever been seen. That's true in the human. So in the human, again, using some of the tools that I talked about previously for Jeremy's project. We could get the same sort of biology if we add the two together, lots of parallel fibers. Again, starting from a system where you had primary stellate cells from the human. Well, let's say that's true. There has to be some novel Wnt then that's triggering this. Because again, they're Wnts made by epithelial cells, immune cells, even fibroblasts. Why would there be this transition suddenly in the cancer stage? So we went back to single cell for all the caveats that I listed before. And we asked, can we see the Wnt and the Wnt receptors and the various enzymes that make Wnt relevant? And it turns out when you look at pericytes, immune cells, fibroblasts, epithelial cells, in the epithelial cell state, you see a lot of porcupine, which is the palmitoyl transferase that sticks a palmitate on the Wnt before it's secreted so that it works. And you see some dots you don't see before, like Wnt7b, Wnt7a. Sort of, you only see them here. There are other Wnts that there are overlaps between the various populations. The Wnt7s are unusual Wnts. They're not studied a lot, but we looked into them. And in this case, the in situ probes are orange, and you see the Wnt7s in the cancer cells. And when you do, you see the baby blue cells, which are, we call them Wnt sensing calves right next to them. And in the cancer, it's everywhere. It's ubiquitous. And if you look in humans, it's similar. You see the ligands in the cancer cells and the baby blue cells in the stroma. It turns out in the Pannin state. So again, I said before, Pannin, there's millions in the mouse. They don't all progress. So if you look hard in the mouse pancreas, you'll have Pannins that are negative for Wnt7, and you never have the baby blue cell next to it. And you'll have some positives, and they always have the baby blue cell. So the correlation is strong of, finding those two events together. So we said, let's do some genetics. Turns out if you knock out Wnt7, there's no cells. It's actually a required for vitality, survival of a mouse organoid cell. Same is true in the human. So you have to get more clever. And so we decided to make a clip out of exon 4 where we would splice out a several dozen amino acid structure responsible for binding the co-receptor for Wnt7, which is called REC. REC binds to GPR124, which is a 7 transmembrane spanning protein. And all of that then gets ligated to the classic frizzled receptor complex to drive Wnt signaling. So we could make the hypomorph of Wnt7 cell survived. Obviously, that means Wnt7 is binding to some other thing and doing some other event to keep the animals, to keep the cells alive. Because we had a lot of viable cells, we could do real experiments. So you could transplant them and see what happens. So the transplanted one with the Wnt hypomorph in the epithelial state don't make stroma. And they don't make baby blue cells, but they are cancer cells. They're still proliferating and all that. And you can do a kind of reciprocal experiment. You could knock down the co-receptors in the fibroblasts and then ask what happens if you co-culture them and all that stuff. And if you do the Eddy experiment looking for anisotropy and you knock down, in this case, knockdown, old-fashioned knockdown, so not knockout, you're just suppressing 3 different knockdowns. It basically goes from a hairy thing to less, and you're essentially not making more fibers, you're making less. You are suppressing the production of matrix in vitro. What if you did the opposite? What if you blazed Wnt7, turn it into a promoter? Well, when you do that, you make a lot more fibers in that experiment I just talked about, and you can measure it in different ways. This is the mouse Wnt7, which is Wnt7b, and you can do the same in the human. This guy's, you can put the human version. Humans express 7a, mouse 7b as the predominant one. You get basically the same biology. What about the Wnt7b mouse into the mouse? Not knocking it out, giving more. Well, now you make a ton of stroma. Like you're making a lot of stroma now when you do this transplant. And you have a lot of these baby blue cells. The really fascinating thing about this experiment to me still is that the proliferation rate of the cancer cells here is the same as here. And in vitro, the same. So you've jacked up the expression of Wnt7b, but it doesn't affect this in a cell autonomous way, the system. Makes desmoplasia, and you're like, well, so what? Well, these mice, they die a bad death. They get like the most metastasis that we've seen to the liver, to the lungs, da, da, da. And if you look at the microenvironment around them, they're all that stuff, the wints, you know, the baby blue fibroblasts and all that stuff. Well, can you do anything about it? So I talked about the palmitoyltransferase, and there are drugs that block the palmitoyltransferase. They've been called porcupine inhibitors. There are fancier names, and there are actually fancier drugs. But the one we can get our hands on, when we give it to the mice that have the precancer, that have the baby blue cells and the Wnt7 and all that, they lose their baby blue cells. Again, you're treating a whole mouse. So you're porcupine treating the mouse, you're going to cause trouble throughout the mouse, but they could tolerate it for 2 months and you have less desmoplasia and less pannins and all that. So it's an anti-cancer effect in the precancer setting. What about when you have an invasive cancer, the KPC mouse, you see the tumor by ultrasound, do the same trick? Well, so we did that only for a week because the mice only live for 12 days. And here you just see a bunch of cancer cells. Here you see some cancer cells with some red dots. These are red nuclei because we're giving doxorubicin, also called adriamycin, drug we use to treat women with breast cancer. We're using it as a tracer dye to see, could we get more dye in when we give the porcupine inhibitor? And the answer is, yep. And then we could ask, well, do the cells care? And this is a different mouse experiment, no adriamycin, in this case, just gemcitabine, which is a monotherapy nucleoside analog we use for patients. And in the setting of just giving the gem versus giving the gem with having a week of this porcupine inhibitor, you get a lot more DNA damage in the cells, and the cells are, you know, less happy. We don't have a quote survival experiment yet with this scenario, but it looks like we're getting more drugs in by, you know, going after this in a very crude way, which is this, you know, pan-Wnt inhibitor. But the bottom line kind of take home from this is that we think Wnt7 is actually a switching event from a pre-invasive indolent to a pre-invasive aggressive cancer in the mouse duct. And we think it triggers through myofibroblast formation and myofibroblast specification into a Wnt sensing and potentially a tension regulating subtype of fibroblasts. We think this is very separable from how the hedgehog pathway works. Won't have time to talk about that because I'm probably over time already. But we do think that this is possibly a therapeutic target. I was never in the Wnt field before. It's kind of a smaller field than the RAS field now. But I talked to all the developmental biologists who started all those companies and, you know, et cetera, because there's like no good reagents to study this, which stuns me. And no one's working on this. No one's working on, you know, Wnt7. You guys seem to start companies fast. It's a good one to start a company on. But anyway, we think this is actually this is going to be a real target. And so we have these new targets to think about. I think acute inflammation recruits stellate cells to become myofibroblasts because of TGF-beta. And the myofibroblasts, you know, surprisingly secrete axon guidance cues, particularly NGF, but there are others. And we think this is maybe a targetable entity, maybe targeting TGF-beta. There's much better drugs for that today. Going after the NGF axis because there are TRK inhibitors approved for cancer patients. And of course, alpha-1 blockers. So these are all possible. We have no data, but that's what we're trying to pursue. Then the last story, of course, was about Wnt7, which we think is involved in that switch from indolent to aggressive precancer. And again, I just don't have time to talk about it, but those cells that express Wnt7, they express all those progenitor cell markers that Scott Lowe and others published, nestin and all that stuff that they think are the milieu of a cell before it invades. Okay, so the last 3 slides are for Ms. Talbot. You stayed with me the whole time. So what's worth working on still? I think, again, this problem of why does only 1 out of 50 become pancreas cancer is a really important problem. And there are going to be likely lots of different explanations. But trying to understand that is very important. And so if you're at that stage where you can shift gears, think about that one. The second point is, how can we diagnose pancreas cancer early? So not the biology, but is there better ways to diagnose it? And last night I talked about studying pancreas health as one such approach. Obviously, if you really had the ambition, you could work with Dr. Drake and actually figure out better glycosylation markers that you could use for identifying pancreas cancer biomarkers. There's all kinds of other ideas. We don't know what's gonna work in this space. If you have the capability of working with an imaging lab, I think imaging is going to get much better than it is today. We do CAT scans, we do MRIs. That's not going to work. It has to be at a cellular level. So it has to be probably an activity probe of some kind that a few cells, or let's say 1,000 cells, would be good enough to give you a signal. Likely would have to involve radiation. But again, this last point was something I talked about last night, pancreatic tissue vitality. Could we prevent pancreas cancer? So 1 out of 50 people get it. What if it was, 1 out of 500? That would be great. Well, how are we going to get there? Well, first, let's figure out why do certain people get cancer in the first place? Most of the people who have a family history of pancreas cancer, we don't know the gene. Vast majority, that's the case. Turns out that's true for breast cancer too. I was surprised. I learned that just a few years ago, and I thought, why didn't I know that? I thought BRCA, P16 kind of explained most, not true at all. So there are other things that explain why people get pancreas cancer if it's inherited. If I were young, I would work on this. I think it's probably related to inflammation. Maybe not even inflammation in the epithelial cells that have the RAS mutation. You could say some version of pancreatitis. Maybe it could be some version of immune problems, because immune cells do live in your pancreas and they come and they go, but you also have resident monocytes. And so this is an important problem, and I think deserves more attention. There are ethnic groups and racial groups that get more pancreas cancer or worse pancreas cancer than others. I don't think we understand that at all. And these days it's not PC to talk about that, but BFD. We should study it because we're going to learn things by studying different populations. And so it's very important to think about. Actually, the worst pancreas cancer in our country are the native Hawaiians. And I actually went out there to try to help them set up a system, because I was trying to help them learn how to make organoids, and they couldn't send their tissue to New York because it was like a tribal thing about the tissue can't leave the island. I'm like, fine, send your people. And so I went out there, I talked to them, and I thought in the beginning it was just because of obesity and all, and bad diet, because our diet imposed upon them. It's not that, it's something else. But no one's working on it, and maybe they're the key to understanding this. I'm partly Scandinavian, and the Scandinavian countries have a lot of pancreas cancer, but I think it's because we just drink too much and all of that. But I think there's something interesting going on in Hawaii, and of course Africans have more and they do worse. Hispanics don't have more, they just do worse. And you could say access to care, and maybe, but it might be something else. And remember the first vignette about the sympathetic nerves? Other people would say that's stress. Right? When people say stress and cancer, that's real. That's biological. Sympathetic nerves are all about stress. Okay, so anyway, the third point, exposures, mutagens or inflammagens. When people who smoke have more pancreas cancer, why? If someone can answer that, that's an important finding. And it's not mutations in the pancreas cancer genome, because they haven't found them. All right, so what is it about smoking that increases the risk of pancreas cancer? I think a lot of it's going to funnel back to inflammation. And then, of course, we want to prevent if we can. And so there are interesting ways to do so with these new drugs against RAS and new vaccines against RAS. And this is the last teaching slide. We need to treat our patients better than we are before. We need to change the way we do clinical trials. I talked about that last night. I know the medics have got up to go see their patients, which is important, because I'm a medic too. But this is really important. And this is where the scientists could help the doctors design better ways to do trials. We have these new drugs called RAS drugs. They're going to be a big deal. They're not going to take your career away. They're going to make your career 10 times more important, because they're not going to cure anybody. They're just going to kick the can down the road And then there are going to be more people on the road because of that. And so thinking of ways to take that door opening moment from the RAS drugs is going to be very important. That's where science is going to play a big role. Maybe the fibroblasts are going to play a role in that. I don't know. And I've already talked about the continuous enrollment on trials, and we talked about the systemic illnesses a little bit. So these are the folks who made the discoveries. Juicy and Mojdeh made the Wnt7b discovery. Sandeep is the guy who figured out how to get us normal fibroblasts, and Jeremy made the sympathetic nerve invasion into the pancreas observation. I'm over by probably a lot. Sorry about that, but I can take questions. Thank you.
Question. Coming over to you. You asked a question last night. All right, you can ask him that.
Hi, Dr. Tuveson. Great talk. Really interesting work. I also didn't think about Wnt7 before this, so that's cool. I'm wondering how your Wnt ablation therapy compares to something like a pegylated hyaluronidase. I understand that it also targets the MyCAFs, but what do you think it would do differently that would make it better? Because that trial failed. And I guess I'm just trying to understand if there's a combination that we can use that would make it more beneficial.
So good question. So hyaluronidase, particularly the pegylated version, in the mouse model, enables more drug to be delivered to the pancreas tumor and prolongs the survival of the mice by 3 weeks. So instead of living for 12 days, they live for 36 days, like that, medium survival. So actually it was pretty effective in the mouse. When it went into the clinic, some of the early phase trials, even a phase 2, looked promising, but it totally fell flat in a randomized phase 3 with FOLFIRINOX. And if you talk to the doctors who did the trial, it turns out the patients that were on the PEG with the FOLFIRINOX got a lot less FOLFIRINOX than the patients on the FOLFIRINOX plus placebo, because the side effects from FOLFIRINOX were exceedingly high in the PEG-treated patients. So you would ask, why don't you do Gem-Abraxane with PEG-PH20? But when you're a company, you basically have one shot. So they're out. Yeah, they're out. So this is the unfortunate thing about when the private sector thinks it knows enough to run everything. The private sector is important to make the reagents and to sell the reagents. The design of the trial should be left to the people who are the experts. That's just a generic point. Many of you have your little companies. Make damn sure that you play a control. You want to fail yourself. You don't want someone to fail on your experiment because they mess it up. I was, what is today? Friday. So yesterday morning, I'm in Manhattan talking like a room like this, a bunch of, V whatever, C or P, capital or philanthropy, which is fake term. People who want to like do this, who want to try, start a company on this, that, and the other thing. And they wanted me to talk about the Valley of Death. Maybe some of you have heard about the Valley of Death. I don't mean Death Valley, the Valley of Death, which is this term which has been invented by us that describes how the academic discoveries don't translate into clinical reality. In fact, our government has now embraced these terms and have come up with new approaches in modeling because of it, and new approaches and methods because of it. So the Valley of Death, I explained to these very distinguished and I'll just say distinguished people yesterday. I call it the Valley of Ignorance, because when trials are designed and interpreted, they almost never have what I would call the controls. They don't have the tissue. They don't know if their drug is in the tumor. They don't know if the target is engaged in the tumor. They're like hoping. I'm like, don't hope, know things. And the same was true of the hedgehog trial that was done, 16 years ago. The company called me and said, ugh. I said, well, what does the tissue say? And what tissue? I'm like, what kind of idiots are designing these trials? And again, I'm a medical doctor. I know what it takes to do things, including write trials. So again, back to your drug delivery point, I don't think we have an answer yet to that trial, and we won't get one. Therefore, if we ever get involved in this one, I want to make darn sure that we have tissue. so that we go in eyes wide open. Now, hyaluronidase does not do anything to the axes I talked about. It just opens the vasculature because it depresses the pressure in the tumor. This would do it differently because it would actually prevent the construction of the entity that creates the barrier, if that makes sense. Okay. So, but good question. And if you study drug delivery, don't give up.
So do you think that they also provide some sort of like diacylglycerol or something to the cancer cells to promote their growth?
Well, I mean, maybe, but the DAG is going to be produced in this case, inside the fibroblasts. So it would be, you know, activation of PKC and other things inside the fibroblasts, which quite possibly is part of their biology. So yes, I suspect it is a big part of it too. We just haven't looked at. Great.
Secrete the diacylglycerol.
It could do that too. Yeah, no, I absolutely.
We measure, by the way, we have lipidomics facility here.
You know, lipidomics is hard to do. So I should talk to you. You have glycomics and lipidomics, two of the hardest things to do. So thank you for that.
David, thank you for sharing some tremendous talks with us. Thank you, everyone, for coming today.